Hello, uh, Genetics CBio 312. Here we are at Clark Atlanta University, and I want to talk to you today about epigenetics, or the emerging field of epigenetics. Epigenetics is really at the forefront of science being researched heavily right now, as not everything is completely clear as to how epigenetics works, but that's why it's such a hot field of study, is that there are a lot of unfinished questions to be answered in the field of epigenetics. So what is epigenetics? Epigenetics is the study of heritable changes, so phenotypic changes that happen in organisms through the generations that are correlated not to changes in the genetic sequence, but to changes in epigenetic marks on top of that sequence. So epi comes from the Greek on top of, genetics, so things on top of genetics. We'll get to kind of more of the history of the term a little later, but I want to just start with the first story in the chapter 21, and go to a town in Sweden called Överkalix. So now we're traveling across the Atlantic Ocean to a little town north of the Arctic Circle in northern Sweden called Överkalix, where in the Middle Ages the townspeople kept extremely precise records of both their crop harvests, the amount of food that they harvested every year, and then also the health status of all of their citizens in this small town. And so what that has allowed modern researchers to do is go back and look very closely and correlate the diets of children raised in times of plenty, times of lots of food available, and their health, and then their children's health, and then their children's health, so their grandchildren's health, and then compare that to people who lived in around the same time, that grew up and developed in times of starvation. So when there was hunger winters because there was harsh winters and poor crop yields, what did the health look like of those people, and then their children and their children's children? And what's really interesting is when we look at the records in Överkalix, we see that grandchildren of people that were raised during a time of plenty were actually 4 times more likely to develop heart disease and other health problems, risk of developing diabetes, etc., than people who were going through the crucial part of development in times of starvation. That's really an interesting observation. And so because of those correlations happening over and over throughout the generations, they've kind of attributed the health outcomes of the grandchildren to the diets of the grandparents. And so here we see an example of what appears to be transgenerational environmental impact on the health of an entire family. So this brings up this huge question of, well, how is that possible? How do chemical environmental exposures get transmitted through the sperm and the egg twice, basically, from a grandparent to a grandchild? So that's what we're going to explore today. What is epigenetics? This particular example, and then a few other examples of epigenetics as it's been observed so far, and what the current state of research is on these things. So the term epigenetics actually was coined by this man, Conrad Waddington, in about 1942, and it was his intention to merge the words epigenesis, which basically means embryogenesis, how an embryo develops into an adult, and genetics. And he was trying to encourage a kind of genetic understanding of how a single cell with identical genetic information can turn into a various array of different cell types throughout development. Um, as the field has moved forward, though, the term epigenetics has become more narrow and kind of now means and is correlated to these chemical modifications that happen on top of both DNA and the histone proteins that we'll talk about later. In this image by Conrad Waddington, you see his version of cell fate determination being represented here by this being representative of a cell, a single cell that's in a stem cell state or pluripotent, meaning it can go any of the different ways, kind of rolling down a hill and differentiating. And as it comes to the bottom of any one of these troughs or valleys here, it then becomes its dedicated cell type. And so this kind of still is a crude approximation of how stem cells kind of find their fate as the different cell types. And we're only now coming to understand that changes to the chromatin, to epigenetic changes to both the DNA, methylation, acetylation, ubiquitination, all these different modifications that happen on top of the genetic material are what cause the differential gene expression that then leads to a cell acquiring these different fates. Something that's even more recent is this idea of induced pluripotency, so pushing the cells back up the hill and getting adult fully formed cells like fibroblasts to de-differentiate back into these pluripotent stem cells. So several different changes to chromatin structure that are not changes to the sequence of DNA can contribute to this kind of cell fate determination. These include changes to chromatin structure that in turn leads to changes in gene expression, as we've seen before. As if you've got a region of the genome that's tightly compacted, you're going to have less access for the transcriptional machinery. Therefore, those genes that are tightly compacted are kind of shut down. Whereas as chromatin structure is loosened, then you have more access to the transcription machinery, and therefore you have more gene expression off of the more loose or euchromatin. So compacted chromatin is called heterochromatin. Open chromatin that's more easily accessible is called euchromatin. And then other molecular mechanisms also alter chromatin structure, such as DNA methylation, and we'll go into exactly what DNA methyl groups look like when they're added to double-stranded DNA. Also, a whole series of different post-translational modifications can happen to those floppy histone tails that we were talking about before. These include methylation, acetylation, ubiquitination. There's also glycosylation. Lots of different things can be happening that affect the overall structure of a region of a chromosome, and that affects its accessibility by the transcription machinery. The other interesting thing, and we'll get to this when we talk about X inactivation, so you know about the X chromosome is the chromosome that's sex-linked for females. When you have two X chromosomes in a female, you need to shut down one of those, and we'll see that RNA molecules are kind of responsible for the total compaction of the X1 of the two X chromosomes present in females. So let's talk here about methylation of cytosines. So often cytosines are methylated on their 5' carbon. Here you see the pink methyl group, and not all cytosines are always methylated. Usually CPGs, meaning cytosines connected by a phosphate group to a neighboring guanine, are methylated on their 5' carbon, and you see here what that does. It changes how this major groove of DNA looks. So if you can imagine a transcription factor or a protein that goes and ought to bind to a certain region of DNA, if that DNA has been methylated at a cytosine, it's not going to be able to recognize that region like it would be if these were not there. And so they act as kind of little blockers, little road blockers on the DNA sequence. Yeah, so these pink things are the methyl groups, and they kind of change the structure of the major groove of DNA such that transcription factors can't get in there and bind. So how does methylation, if it happens kind of on opposite sides of a double-stranded DNA molecule, pass through a round of replication? Well, this is really interesting. So here we see a piece of DNA that's methylated on both sides of the DNA strand. Now, it gets replicated, right? So the red here represents the newly synthesizing DNA strands that are copying this already existing DNA strand. Now, what you get, because of what we learned from the Mendelssohn-Stahl experiments, is you get this semi-conservative replication that happens, right? So you've got gray old template strands and then red newly synthesized strands of DNA, right? So what happens here is you get these methyltransferase enzymes. These are these green things right here. And the methyltransferase enzymes, they recognize these hemimethylated DNA cytosines in the DNA, and they methylate the opposite strand. So these are called maintenance methyltransferases because they're actually maintaining the methylation pattern on the parent strands of DNA through replication. I just think that's fascinating. So a really dramatic example of epigenetic changes that you can see with your eyes are the physical differences between a queen bee and a worker bee. If you look at this queen honeybee on the left here, and you see how much larger she is compared to this worker bee, realize that these two organisms have identical DNA. Their DNA is identical. But because this bee, the queen bee, was chosen to be the queen bee, she was licked and given this special nutrient called royal jelly. And that royal jelly is filled with nutrients and things that actually turn on different transcription factors that then make the same DNA that's in this smaller worker bee turn her body into this much larger mating phenotype, which is very dramatic. So it'd be like if these two were twins, and then the one was given, you know, the best education money it could buy, and the other one was just completely ignored. I mean, you're going to have some major changes in the phenotype of organisms that are treated differently and exposed to different environmental cues. So we already talked about some of the molecular processes that can lead to epigenetic changes, and the ones that I mentioned were methylation, but then also post-translational modifications to the histone tails, and those can be ubiquitination, acetylation, methylation, and all of those kind of are correlated with either active or repressed transcriptional states, so either compacted chromosome region or open chromosome regions. But a lot of those histone modifications are context dependent. Another example of a molecular process that can lead to these epigenetic changes are RNA molecules. And these are X inactivation by this XIST gene, XIST, and paramutation in corn by these small interfering RNAs. So a paramutation is an interaction between two alleles that leads to a heritable change in the expression of one of the two alleles. But this is based on not changes in the DNA sequence of the two different alleles, because often the two alleles in a paramutation are genetically identical, but the accessibility because of transcription factors or small interfering RNA that's associated with the kind of promoter region or some regulatory region of the gene is slightly different in one allele versus another. And if you just associate with that repressed or that activated allele, the other allele then becomes either activated or repressed, and then that change to it to its transcriptional state is itself the heritable thing, not not the sequence itself. So there's examples of paramutation in corn, and then also another example in mice. So when we look at the phenotypes of a mating that happens with paramutation influencing the trait, we see in this corn example, you've got a gene B1 and then B prime that both code for a pigment gene, a pigment protein. And in the B1 homozygous, you see this dark pigmentation, this dark purple pigmentation, and then in the B prime, you see kind of a lighter pigmentation, not nearly as dark purple. But then when you cross them and you get what should be a heterozygote, instead of getting a heterozygote, you get this reversion of the B1 gene into this B prime star gene, meaning that the gene itself in both this case and this case are exactly the same. The sequences are the same. It's the transcription that's changed between one allele and another. And so when you get them crossing, this is the definition of paramutation. You actually change the transcriptional activity of one of the two alleles. And so in this case, we've got the phenotype for the less pigment coming through in the F1 generation, and that's because this B prime has kind of induced the same kind of transcriptional state on its B1 counterpart. Now if we look down at the molecular mechanism of that phenotypic state we just observed, we see that in the B-1 gene, you don't see very much of this modification happening upstream to these tandem repeats. So you get open transcription of the B1 locus. So this represents that dark purple pigment being expressed at high levels. But in the B prime allele, because you've got this modification that's been happening up here in this regulatory region, these tandem repeats, you see that the thickness of this arrow is much less than the thickness of this arrow. And so in the B prime gene, you get less transcription off of the, see how these sections are both blue? In both cases, the sequence here is the same. It's just transcription rate that changes between the B-1 and the B prime. Now here's paramutation. This is where paramutation comes in. So paramutation happens when you get the two alleles coming together. So now you got the B prime and the B-1 allele coming together. And because they're in the same area, we don't really understand the mechanism of how this works, but somehow you get this paramutation in which that modification that was just relegated to the B prime actually shifts over now and affects the B-1, therefore reverting it to a less transcriptionally active state. And so this B prime star is actually this B1 gene that's been paramutated to not allow as much transcription, just like the B prime. So we don't exactly know how this works, but somehow a small interfering RNA kind of comes off of the paramutating gene and kind of deposits on the paramutated gene, in this case B prime to B-1, reverting it to a less transcriptionally active state. In another case of paramutation we see in these mice, when you have the KIT1 plus and the KIT1 plus homozygote, you have a mouse with this solid color, no white tail, no white feet. But when you have a KIT plus and a KIT T heterozygote, you have these brown mice, but they have white feet and this white coloration on their tail. Now you mate these two. This is a homozygote KIT plus and then a heterozygote KIT plus KIT T, and you get half of them which are homozygous for the KIT1, and half of those are wild type, so they don't have the white feet. But then half of them also have the white feet, even though they have the genotype for the KIT plus homozygote. Their phenotype is like the KIT plus KIT T. So this is one of these cases where paramutation violates what we'd expect from Mendelian inheritance, in that the genotype of this mouse right here is KIT plus KIT plus, and so we'd expect its phenotype to reflect its genotype. But because of paramutation that we just explained in the slide before, you do get this this passage of the KIT plus KIT T phenotype in the KIT plus homozygote. So this is an example in which you have the passage of a phenotype that's coming not because of the passage of a gene, but because of a change to a gene. So this is what epigenetics is, and this particular case of epigenetics is called a paramutation. So whenever you have a change in expression of anything that's heritable, that isn't related to the DNA sequence, it's epigenetics, and in this case, paramutation. So one of the most fascinating experiments that has been done in the field of epigenetics in the last 5 years has been this maternal care study in rats. So what this study has shown is that rat pups who are nursed and treated with affection by their mothers as young pups grow up and are less fearful and respond less to stress in their lives, whereas pups that are raised by mothers who neglect them, who don't do this licking and grooming when the pups are a very early age, they are not as well suited to cope with stress and are more fearful as adults, which is really interesting and has far-reaching implications for human beings. Basically showing, and the really interesting thing is genetically identical pups that were either nurtured and licked and cared for intensely or neglected. The genetics could be exactly the same, but the environment was the thing that impacted their epigenome. So the researchers studied the methylation patterns inside the brains of the pups after they had gotten to adulthood, and showed that there were differences in the methylation patterns around the glucocorticoid receptors, which is a stress hormone response element in the pups that were groomed a lot as infants versus those that weren't. So this shows, actual evidence that there are epigenetic differences brought about by environmental differences that happen early on in life. And so this has huge implications, especially when it comes to things like cycles of violence in human human development. If a child comes from an extremely violent household, they're epigenetically perhaps primed to be stressed out for the rest of their life. And so we're still trying to understand and verify that this is true. But the other thing that this has implications for is post-traumatic stress disorder. There's another study that was done last year by a lab at Emory by a man named Kerry Ressler and his group, and they were studying mice. So basically, the Ressler group trained mice to be afraid of a certain smell, acetophenone. And so whenever they would be in their chamber and be exposed to this smell, they would have a little foot shock. Okay, so they're trained to be afraid of this smell. Now, they bred these mice and raised the progeny in a lab across campus away from the parents, and they observed that the mice who were the children of this the mice that were trained to be afraid of the smell also had increased levels of expression of that smell receptor and were also afraid of the same smell without having any training by their parents. It was purely epigenetic, so it was transgenerational. Now we don't know the mechanism by which the fear of that smell was passed, but the observation is there, and they did that through bisulfite sequencing, which I'll explain later, of the gene for the receptor for acetophenone. So the fear smell, the gene for the receptor for that smell was actually methylated differently in mice that were the progeny of parents who were trained to be afraid versus those that weren't. Very interesting. So there's a lot more to explore in the world of epigenetics, and we're going to continue to go over some of this in class and in recitation tomorrow. But and I encourage you to read the entirety of chapter 21. But I just want to close here with this explanation of how bisulfite sequencing works. Bisulfite sequencing is basically a mechanism by which you can do DNA sequencing and tell which residues, which cytosine residues were methylated and which were not. And so in any genome, you're going to have certain cytosines that are methylated, like these with the little red dots on top of them, and those that are not. Okay, so so in order to do bisulfite sequencing, you need to have a bioreplicate. So you need to have one sample that's not treated with bisulfite and one genome that is treated with bisulfite. So when you treat with bisulfite, the bisulfite actually catalyzes a reaction where it deaminates the cytosine and actually turns the cytosines to uracil. Okay, so when that happens, then when you go to sequence it, that uracil actually shows up as a thiamine, because in DNA, uracil is synonymous with thiamine. And so then you've got to use those computer algorithms that we were talking about before to compare the sequences. And you're going to see the sequences are generally the same, except for they're going to differ between C's in the untreated and T's in the treated. And so anytime there's a cytosine in the untreated and a thiamine in the treated, at least in the output of the sequencing results, then you know that that cytosine had been methylated. And so you can kind of map out the methylation pattern along the entire genome. Now this is part of what we call the epigenome. So that's the goal now of a lot of research, is to map out the entire epigenome. So you've got the sequence of the entire genome. That's one bit of information. And then if you really want the whole picture of how the genome is being regulated, which genes are on, which genes are off, you have to do the epigenome. Now you can imagine that the epigenome could be different for every single cell in a human body. Now it makes sense that certain cell types that are the same should have similar epigenomes. And that's what my PI, my boss, Roger Deal at Emory, he's looking at trying to isolate the nuclei of one cell type at a time from plants, and then look at the epigenome of one cell type at a time. And so that's where we are in the process of understanding epigenetics, is we're actually sorting out individual epigenomes for individual cell types. Very interesting as well is, what does the epigenome of a cancer cell look like versus a healthy cell? And what you see is there are massive changes in methylation patterns, histone modification patterns, and things like that. So I hope that this has given you kind of a primer on epigenetics and hasn't overwhelmed you. I'm going to leave it here, even though there's lots more to talk about, and we'll go in more detail into how X inactivation happens. But I just wanted to put this out there just so that you have kind of a primer and kind of getting you launched into reading chapter 21, which is a brand new chapter in this textbook. So this is not only my first time teaching this chapter, but this is the world's first time teaching this chapter, because epigenetics is a really new field. Anyway, yeah, I hope there's some useful information in here, and if you have any questions, email me. You all have my email address, and I'm on Canvas now, so we can talk through Canvas. Message me on Canvas. Okay, have a great night, and we'll see you in class.